Let us continue our discussion on chapter 3, Human Reproduction, class 12th NCERT NEET Biology. And this is human reproduction, and the specific topic that we will continue to discuss would be 3.3 gametogenesis, and this video will be on the gametogenesis in the human female, which the process would be called as oogenesis. So you know that in the female the gamete is called as the ovum or the egg, and the process of the formation of the ovum or egg would be called as oogenesis. And if you contrast it with the process of the spermatogenesis, then you can get the etymology correct. So I hope this is absolutely clear. Now let us begin the discussion very fast. The first important question is that there is a fundamental difference between the two processes. I told you that the human males, the process of the spermatogenesis begins with the onset of puberty, that coincides with the secretion of or the release of GnRH by the hypothalamus. Though it is true that in the female, the development of the secondary sexual character or the onset of the puberty actually occurs because of the release of GnRH only, but there is a difference in the process of the gametogenesis. The gametogenesis in the female sex actually begins before the birth. So one more important point that just came to my mind is that the onset of the puberty in females is 1 to 2 years earlier than the onset of puberty in males. But we were talking about the process of gametogenesis. The fundamental thing to understand is that in the human females, the gametogenesis or the process of oogenesis begins before birth. This is in striking contrast with the human males, where the process begins at the onset of puberty. This is an important thing to understand. Now, what happens during fetal life? So what happens during fetal life? So this is ovary, and we are saying that in the fetal life, what happens is that some of the cell, we are not going into the unnecessary details. So I am limiting myself largely to the discussion of the concepts and the facts that is given in NCERT. Again and again, I am telling you that the NEET entrance exam would largely be based on this syllabus of your NCERT textbook only. And mind you, this is our last revision before the entrance examination. So we must be very prudent. We should be very clear about what to read and more clear about what not to read, which is probably more important for the success in the NEET entrance exam. So there in the human male, we talked about spermatogonia, and here we will talk about the oogonia. So the corresponding cells would be spermatogonia and oogonia. Spermatogonia in male and oogonia in the female. Now, this oogonia will differentiate into, remember the spermatogonia ultimately divided and differentiated into the primary spermatocyte, and here the oogonia will divide and actually they will migrate into the cortex of the ovary and will get differentiated into what is called as a primary oocyte. So we are beginning the story with a primary oocyte, and like the primary spermatocyte, the primary oocyte in the female would also be a diploid cell. Now, please remember a very important fact here. So if we look at the reproductive period of the human female, that is from menarche to menopause, onset of the menstrual cycle to the cessation of the menstrual cycle, that would be the reproductive period in the life of the female. So let us say this is roughly 400 months. So these 400 months, in every month she would be releasing a single ovum in each menstrual cycle. That would be roughly 400 or maybe 450 ova in the entire reproductive age. Now this is the number we are talking about. Just contrast it with the number in the human males. In the human males, we said that this number in each ejaculate can be 400 million in one time. So we are talking about a period of, let us say, a period of around 30-35 years and only 400 ova, and in one ejaculate 400 million. So there is a difference between 400 and 400 million. Now, which means that the oogonia that they differentiate into the primary oocyte, the oogonia are not going to continuously divide and replenish their number as was happening in the cases of the spermatogonia in the human males. All the ova that the female is ever going to produce during the reproductive period of her life will be present in the ovary at the time of the birth of the female child. So when a female child is born, all the future ova are already present in the ovary, though they are not fully developed. We will see what happens at the time of birth, and actually first we will see what happens before birth. So we are saying that okay, some oogonia, actually we say that they actually differentiate into what are called as a primary oocyte, and these primary oocyte we are saying is actually a diploid cell. Now what happens is that this primary oocyte, just keeping the things very simple, will be surrounded by a layer of cells, and this layer of cell would be called as, later on we will call it as a layer of granulosa cells. So we are looking at a primordial ovum. This ovum is actually a primary oocyte. It is surrounded by a layer of granulosa cells. So these granulosa cells will be very important in the production of the steroid hormones, the estrogen and progesterone later on. But just remember, this is a very primordial thing. This is before primary. So this is a primordial follicle. So at the primordial follicle, probably I will not like to call it as granulosa cell. We will call it as granulosa cells later on. But we must remember that whatever is the single layer of the cell surrounding the primary oocyte at the very primordial stage would actually give rise to granulosa cells later. And this follicle, this is the follicle. I told you in one of the videos that the cortex of the ovary would be containing large number of follicles at various stages of development. You can see that the follicle is actually present in the cortex, and we will call this thing as a primordial follicle. Now this primordial follicle, this is actually primary oocyte, and this primary oocyte begins meiosis 1. So as was the case in the human males, the primary spermatocyte underwent the reduction division. So this division actually begins in the fetal life in the females as well. The primary oocyte, I would say, it begins meiosis 1. Begins meiosis 1, which is the reductional division, and is arrested at a stage which is called as the prophase 1. You know, prophase 1 of meiosis 1. And to be precise, we are also aware of the fact that the oocytes can be arrested for months or years in diplotein stage. I have discussed this very important point in cell cycle and cell division. So now we are saying is that this is the primary oocyte. This is still the primary oocyte. It has begun meiosis 1. It has proceeded to prophase 1 diplotein of meiosis 1 and gets arrested there, and it is surrounded by a layer of what is called as the primary follicles. So this is what is going to happen. The layer of granulosa cells, not primary follicles. I am in a hurry to tell you what exactly is a primary follicle. So let me remind you this, that this is a primary oocyte, and it is arrested at diplotein of prophase 1 of meiosis 1. It is surrounded by a single layer of, now we can call this very clearly as granulosa cell, and I am calling this structure as primary follicle. So what happens is that the development of the primary follicle actually begins around second month of the intrauterine life. So second month in the fetal life of the human fetus, this development of the follicle actually begins, and this follicle number actually increases to approximately 7 million by the 5th month of the intrauterine life. So all these follicles, no new follicles will be formed later on. So it begins from the second month, reaches a zenith, I would say a maximum number at around 5 months of intrauterine life, and that number may be actually around 7 million. So this would be the number of the primary follicles. Does it mean that when the female is born, she has 7 million follicles, primary follicles in her ovary? The answer is no. Though all primary follicles will be carrying primary oocyte, please remember this, and they will be arrested at diplotin of meiosis 1 of prophase 1. Now what happens is that there is a very interesting thing which is called as follicular atresia. So follicular atresia. So this is an important point to understand, that there is follicular atresia. Now follicular atresia means that out of these 7 million, almost all of them will undergo atresia. They become atretic and they become very small in size, and we can say they get degenerated. Why I am saying almost all of them? Because I know that only 400 or so follicles will develop or get mature enough to release an ovum. So if you look at the customary fate of a follicle, it should be actually atresia and not releasing an ovum, because out of 7 million we are saying that only 400 or so will actually release an ovum. So this is very interesting, and let me bring your NCERT right now. So we are saying that these primary follicle, they get degenerated, and atresia begins from the 5th month onwards during intrauterine life, gestation period only. So from the 5th month, it actually rapidly decreases, and this continues till the end of the reproductive period. So that means sometimes the follicular atresia may occur at a very rapid rate, sometimes the rate may be slow, but it is inevitable. The number is constantly coming down. And look what your NCERT is saying. It says that at puberty only 60,000 to 80,000 primary follicles are left in each ovary. So there are 2 ovaries, please remember. So 60,000 to 80,000 primary follicles in each ovaries, and we are saying at the onset of puberty, so 1-2 years earlier than the male. So we are talking of an age of around 11 or 12 years. So that is 11 to 13 years. That would be the age where the number actually decreases, falls significantly to 60,000 to 80,000 in each ovary. Just imagine the amount of atresia that is going on. So I hope this is absolutely clear. That means that when the female baby is born, her ovary is carrying large number of primary follicles, and these primary follicles carry primary oocyte, and this primary oocyte has begun meiosis 1 and proceeds to diplotin of prophase 1 and gets arrested. So at the birth, this is the position. From birth till puberty, nothing changes here. So nothing changes here. The primary, actually one thing does change, and that would be the follicular atresia. Apart from that, I am saying that if you are considering a single follicle, nothing is going to change if it is not undergoing atresia, and the primary oocyte will remain primary oocyte arrested at meiosis 1 prophase 1. I hope this is absolutely clear. Now, the further changes in the follicle, the development will occur at the onset of the puberty. And again in females, the onset of the puberty, like males, coincides with the release of gonadotropin releasing hormone by the hypothalamus, and again the gonadotropin releasing hormone, this hypothalamic hormone, will reach the anterior pituitary through the portal circulation and will stimulate the gonadotrophs. And then what is going to happen is that these basophilic gonadotrope cells, they will actually release gonadotropin, and one of them is follicle stimulating hormone, FSH. Now I hope you understand what is going to happen to the follicles. So therefore this FSH will lead to the further development, will actually begin the further development of the follicle with the help of the LH hormone, which is called as luteinizing hormone. We will look at the details of what actually is the role of FSH and SHH is in the process in the next section, which is actually called as the menstrual cycle. But for now, it suffices to say that the further development will occur at the time of the onset of puberty or beginning of the menstrual cycle. What happens is that this primary follicle gets converted into a secondary follicle. So what happens that when it further develops, it gets converted into a secondary follicle, and the structure of secondary follicle would be something like this. So let me take another page. For this. We are saying that this is the primary follicle. It has primary oocyte. So I will take some time in teaching you this. This is a very simple concept, but very very commonly not understood well by most of the students. This is the primary follicle that we are talking about. This primary follicle, remember, FSH-LH, will undergo some change and will get converted into what is called as a secondary follicle. And this secondary oocyte, again we will see later on, it will be surrounded by a glycoprotein layer. A glycoprotein layer, the primary oocyte would be surrounded by a glycoprotein layer, and this layer would be called as the pellucid zone or zona pellucida. So this is zona pellucida, and it would be surrounded by the granulosa cells, obviously, and these granulosa cells have now increased in their number. So they have increased in number. Just look at this. So they are increased in number. And this is what a secondary follicle will look like. I hope this is absolutely clear. And obviously the granulosa cells would be resting on or would be having a basal lamina. So let us take a different color. So I am saying this is the basal lamina of the granulosa cells, and some other cells will actually get associated on the outside. So some islands of the cells will get associated on the outside, and we are going to call these cells as the theca cells. So remember, this is what is called as the theca cells. So if I go for the labeling part, so this is still primary oocyte. So this is still primary oocyte arrested at blah blah blah, and then it is surrounded by a glycoprotein layer which is called as zona pellucida, and then outside the zona pellucida you have the granulosa cells, then you have the basal lamina, and outside there are what are called as, let me write this thing, this is the theca cells. Now what happens is that these granulosa cells, they will start secreting a fluid, and this fluid will get accumulated somewhere here, and this will lead to the pushing of the oocyte that is surrounded by some of these cells to one of the ends of the developing follicle. So remember, this is a secondary follicle. Now in further development, what happens is that this oocyte, we will talk about that what type of oocyte is this right now. So this is I'm just not giving you the name of the oocyte. I'm not saying that this is a primary oocyte right now, because for the obvious reason that we are going to discuss, some changes will be happening here, and this is still surrounded by zona pellucida. And what we are saying is that because of the accumulation of the fluid, there are some cells that will surround this particular oocyte at one end. You can see that this is a group of cells that is surrounding or suspending this oocyte, and there are some cells that will be formed something like this. I hope you got the idea, because what is happening is that a fluid is getting collected. So a fluid is getting collected. So let us make a few layers of the cell. These are the granulosa cells, and we will call these cells with different names right now, but first you understand that some fluid has accumulated here. It has pushed the oocytes towards one of the end, and obviously surrounding it would be the basal lamina. And outside the basal lamina, now we will see the number of the theca cells actually increase. So the number of the theca cells have increased. So there is now a formed layer of theca cells, though not very dense with respect to the granulosa cell, but these are the theca cells. And what you will see that further, the theca cells are surrounded by some connective tissue. So that means this outer part, the part outer to the basal lamina, is now differentiated into two. And we call this follicle as a tertiary follicle. So this follicle would be called as tertiary follicle. This would be called as a vesicular follicle because of the fluid present, and because this fluid is called as the antral fluid, and the cavity and the space that is appearing here, this would be called as the antrum. So this can also be called as an antral follicle. and we know that this is oocyte, this is zona pellucida. We call these surrounding cells as cumulus oophorus. These cells are the cells of cumulus oophorus, and obviously these are the granulosa cells. And this time, because there is an outer layer as well, the theca cells now will be called as theca interna. These magically change names, and then this would be outside, would be the theca externa. So this would be theca externa, and I am not yet commenting on the oocyte, because it is at this stage the primary oocyte completes meiosis 1 and results in the formation of two cells. And what happens is that the primary oocyte, which is a diploid cell, you know this, it is a diploid cell, has 46 chromosome, undergoes meiosis 1. This is the reduction division, results in the formation of two unequal cell. One of the cell retains majority of the cytoplasm of the mother cell. This cell, though haploid, this would be haploid, this would be haploid, this cell would be called as a secondary oocyte. Now this is very, very different from what happened in spermatogenesis. In spermatogenesis, the primary spermatocytes produced two equal secondary spermatocytes. But here what is happening is the division is unequal, and one of the daughter cell is retaining majority of the cytoplasm. The other cell, which doesn't have significant amount of cytoplasm, it would be called as the first polar body. So this would be called as the first polar body. I hope this thing is now absolutely clear. So now what we are saying is that this cell is actually secondary oocyte. The primary oocyte has completed the first meiotic division, has released the first polar body. You can see the polar body in the space surrounding the oocyte. The first polar body has been released, and what we are calling this as, it is the secondary oocyte. Now what happens is that this secondary oocyte actually begins meiosis 2. Now this is very, very complicating. So this is why I tell my students that female reproductive physiology is not easy, and I say now that it begins meiosis 2 and actually gets arrested at metaphase 2. And whatever is released at ovulation is actually this secondary oocyte. So normally we call it as an ovum, but whatever is released at ovulation is the secondary oocyte arrested at metaphase 2 of meiosis 2. And this thing, when gets further matured, so this tertiary follicle that you are seeing, when it gets further matured, this is called as the Graafian follicle. So completely mature follicle is called as a Graafian follicle. The Graafian follicle, remember, still contains secondary oocyte. The rupture of the Graafian follicle will lead to the release of the secondary oocyte from the surface of the ovary into the peritoneal cavity. The process is called as ovulation, and this secondary oocyte would have begun meiosis 2 and will be arrested at metaphase 2 of meiosis 2. And the Graafian follicle has been so named in the honor of the scientist who discovered the human ovum. The name of the scientist was R.D. Graaf, and that is why it is called as Graafian follicle. So you will say that, sir, you say that do not go lot of outside NCERT, but still you are telling something which is not given in NCERT. Yes, the reason is because now you will be able to understand what is written in your NCERT with the help of all the discussions that I have done regarding this very, very important topic. There is a very important question here, a very important question. What is the advantage of the fact that one of the cell retains majority of the cytoplasm? What is the advantage of the one of the cell actually retaining majority of the cytoplasm? So what we will do is that, okay, this is primary oocyte. So let us forget about what is happening when. So we are saying this is primary oocyte, just to look at the complete process. This is primary oocyte, this is diploid cell. We say that it will undergo meiosis 1. Obviously at some point of time it is going to complete the meiotic 1 or the reductional division. It will lead to the formation of two daughter cell. One of them will be retaining majority of the cytoplasm, the other will be devoid of any significant amount of cytoplasm. This haploid cell would be called as a secondary oocyte. and this would be called as first polar body. Now what happens later on? We will see that actually we just now I told you that this actually begins meiosis 2 and gets arrested at metaphase 2. So this is a very economization of the divisions in the female sex. So this division will happen at the time of the fertilization. As soon as the sperms enters into the cytoplasm of the oocyte or the ooplasm, this division will be completed only then. But let us forget about the timing. Sometime what is going to happen is that this secondary oocyte will complete meiosis 2 and again will result in the formation of two unequal cells, and one of the cells will retain majority of the cytoplasm again, and the second daughter cell will not have significant amount of any cytoplasm. This cell would be called as ooted. Remember spermatid in spermatogenesis, and this thing gets matured into, then it is called as the ovum. So this ooted, the further maturation would be called as the ovum, and this thing would be called as the second polar body. Second polar body. Now, before coming to the question of the question that we are discussing, that what is the advantage, let me complete the process. What happens is that obviously this thing will be now called as ovum. What happens to the polar bodies? Now, this polar body may or may not undergo meiosis 2. And if it undergoes at all into meiosis 2, it will lead to the formation of two haploid daughter cells, and these would be called as, look at the word, secondary polar bodies. So please differentiate between a second polar body and secondary polar bodies, the difference between the two. And remember, these polar bodies, there are three of them. If at all it is formed, they will undergo degenerate, then they will get degenerated, they will undergo degeneration. So that means they do not produce any ovum at all. So they degenerate. So what our answer becomes if we are asked, how many ovum can be formed from a primary oocyte? The answer would be 1. What will be the number of the ovum that can be because of secondary oocyte? Again the answer is 1. Ooted, again the answer is 1. The polar body is 0. So for just remember, primary spermatocyte would be 4 sperms, secondary spermatocyte in males would be 2 sperms, and a spermatid would be 1. Here primary oocyte 1, secondary oocyte 1, ooted 1, and polar body 0. Now if at all they have to divide and then degenerate, it does not make sense. So what is happening during evolution, that larger and larger number of the cases we are seeing that this division does not occur, and the first polar body gets degenerated on its own. So therefore this division is not needed. One very important thing. Now the second thing was, the original question that we were trying to discuss is that what is the advantage of this cell retaining majority of the cytoplasm? The answer is pretty straightforward. We have already discussed the structure of the sperm. So sperm doesn't have the cytoplasm, a large amount of cytoplasm of its own. So that means when it fertilizes the ovum, it results in the formation of the zygote. The zygote divides into a multicellular structure which is called as the embryo. These cells have to have some nutrition, and that nutrition will not be provided by the sperm because it does not contain any significant amount of cytoplasm. So therefore this cell actually retains majority of the cytoplasm. Many of the maternal genes are active, the maternal RNAs would be transcribed and proteins will be translated. It will contain the nutrients for the initial divisions that would be necessary, because before attachment to the wall of the uterus there must be a source of nutrition available for the developing very initial embryonic stage, and that would be the purpose that would be served by one of the cells retaining majority of the cytoplasm. Similarly, you can be asked that what would be the purpose of the formation of the first polar body. Remember, this has 46 chromosomes and you need a gamete which has 23. So you will have to have a dumping ground for 23 chromosomes. So where do you dump them? So therefore two cells must be formed. One of the cell will be functioning as a dumping ground for the additional material that you do not require. And that would be the function that would be served by the polar bodies. That function has been actually served here, and this polar body will also get degenerated. So there is no need of this this division, but it is an evolutionary legacy, and but we are seeing with evolution that it is not occurring in many cases. The first polar body itself gets degenerated. So this is the discussion on actually what I would do is that I would just give you a brief overview of the things. So we said that oogenesis actually begins before birth. Then we said that there is something which is called as primordial follicle. We know what are granulosa cell, then the primary follicle, then we know that this has actually primary oocyte. This has begun meiosis 1 and is arrested at the diplotin stage of prophase 1 of meiosis 1. This is the condition at the time of the birth of the baby. Obviously I have talked about the follicular atresia beginning 5 months. It is actually inevitable, it happens all the time. The speed or the rate may vary, and then we said that actually everything happens after the onset of the puberty. And then this primary follicle will get converted into secondary follicle, which will be converted into a tertiary vesicular or antral follicle. This has a cavity which is called as antrum, will be filled with a fluid which is called as the antral fluid. We have cumulus oophorus, and in this tertiary follicle the primary oocyte actually completes meiosis 1, releases the first polar body, gets itself converted into a secondary oocyte which begins meiosis 2 and gets arrested at metaphase 2 of meiosis 2. It is at this stage that the ovulation actually occurs. That means what is released at the time of ovulation is a secondary oocyte arrested at metaphase 2 of meiosis 2. And then we had an overview of the process of the oogenesis, and we discussed what would be the advantage of one of the cell retaining majority of the cytoplasm, and what would be the advantage, if at all, of the formation of the polar bodies. So I hope you have understood this very important video, and you are enjoying this, and this is just before your NEET entrance examination. So what I would say is that if this topic appears a little bit of tricky, so what you can do is that if you are following masterclass in biology, so I have given a lot of questions in the understanding portion part of the masterclass NCRT. You can look at the previous year questions as well, and you are ready to go.